Okay, I think we are recording. Okay, so welcome to the 3000788 Introduction to Computational Molecular Biology. So this is different from the bioinformatics course that you can find in Department of Biochemistry. I opened this class to kind of provide an introduction to kind of first year student who has no experience at all. So this class has no assumption about what you know or what you don't know. We might assume some knowledge about molecular biology, but we are not going to assume anything about omics or computational techniques. So everything is going to be explained in detail. So for this first session, I'm going to talk a little bit about what this class is about. And we're going to talk about computational thinking and how it applies in biology. So many of you might be familiar with kind of what we call traditional biology, where you use knowledge and you use wet lab experiment to study kind of cells, study organisms to get new information. But in the modern day, now that we have high throughput sequencing technology, we have a lot of assays, we can collect kind of more data, and we can use computational techniques to learn about biology as well. Okay. So this class about me, I already asked you to kind of introduce yourself. So let me introduce mine. So my name is Sira Sisawat. I got an undergrad in mathematics, but I also study molecular biology. So my expertise is a combination of using computational tools and mathematical models to study biological systems like protein structure or gene expression in cancer patient or in plants, in kind of animal. Basically, this class will be all about applying computational thinking and computational tools to answer biological problems. And we will touch a little bit about machine learning and AI toward the end, because this is an AI era. So even if we just started, like many of you, probably this is your first introduction to computational topics, but machine learning and AI is everywhere. So we need to understand them as well. So the objective of this class is intended for new students. So if you are already experienced, like from your undergrad or from your research, you might find this class too easy. But hopefully, because this class is going to cover very broad topics, like every omics possible, and from imaging to DNA sequencing to mass spectrometry. So hopefully there will be something new that interests you, even if you probably know some of the areas already. And we're going to focus on both the biological motivation, like why do we invent these techniques? Why do we use these techniques? Also on the how, like how do we analyze the data once we perform the experiment? So we're going to look at both the why and the how. And for those of you who want to debug, who want to analyze data, all the assignment, we have 6 assignments. All of them will be hands-on. You're going to be analyzing something. There will be no, you might have heard about this class from past year. I used to ask a conceptual question, like why does this technique work, for example. But with the coming of AI, like large language model, like ChatGPT, you can get answer to those questions easily. But what is going to be more important to you going forward is how to do things, like how to analyze the data and to get the actual experience, hands-on experience. And in the assignment, we're going to also be trying to use language model and see if it can give us the right answer. And we can critique, we can discuss about what language model tells us about computational biology. Okay, so this is the course structure. Some of you already got this kind of schedule, but some of you might already saw it for the first time. So we're going to have 28 sessions. Right now, we schedule it for Monday and Wednesday. But if many of you have conflict, we can talk about shifting it. So we can vote after this if you want the class to change the time. We are still waiting for the room. We have 6 assignments, so it's going to take up 15% each of your grade. So you have 10% extra that if you come to class, if you ask question, if you talk to me, then that's like helping toward your grades. Okay. For the assignment, because some of you might find computational analysis difficult. So in this class, we allow you to work in groups. You don't have to work on your own. You can work with your friends. I only ask that you write your own answer. You can work together, use the same software, but once you go back and put the result into writing, please do it on your own. Don't copy and paste your friend because I'm gonna be able to see it anyway. And even though AI can do a lot of things for your benefits, please don't abuse AI too much. You can use some, you can ask AI some question. And please credit if you ask AI to do anything for you, but don't abuse it. Like don't just ask it to do the entire assignment. Now that you are a graduate student, grade is not that important. What's more important is what you learn. So I'm happy to give everyone an A if you all kind of focus on doing the assignment and ask me if there is any problem. So basically, you can even ask me for hints on how to do the assignment if you think that it's kind of difficult. I don't mind that. It shows me that you are interested in knowing what to do, how to do it. Okay. So for those of you who are advanced, like you have some experience, I can recommend further kind of self-study resources that you can use. First is the courses that I have taken from MIT OpenCourseWare. So Foundations of Computational System Biology. This is a more technical take on the topic that we are going to learn in this class. In this class, we might talk about some algorithm. We might talk about the motivation, but we are not going to like go into the actual detail of every step in the algorithms. But if you are interested in those details, I can recommend you to this class. And for those of you who wants to know more about how machine learning and AI comes into the study of human disease, human genomics, this class by Professor Manolis Keris is highly recommended. Okay. And if you are into programming, like some of you mentioned that, I recommend going to Kaggle if you are new to Python. They have an introduction to Python, introduction to machine learning, introduction to programming that you can study. And they have code that you can write, and you can see the result right away, so you know if you got it wrong or not. It's kind of very interactive. And for those of you who want kind of even more practice of Python and application in data science and computational thinking, these two classes, 6001 and 6002 from MIT OpenCourseWare, I highly recommend it. I already went through the content, and I have recommended this to many medical students and many biological students who want to pick up kind of Python and programming skills. And so far, seems to be working very well for them. These two courses are for first year undergrad student. So it's geared toward student who has no experience. So you should be able to kind of access this. Okay. So I mentioned a lot about computational thinking. So let's talk about what it actually is. Like, have you been using it already, or is it something that is totally new, a new way of approaching the problem? So usually, if you are from biological science, we are in what's called a knowledge-centric field. We rely on past theory, we rely on past knowledge, like which gene does what. The central dogma, like we rely on knowledge in order to design the experiment that we will be doing, in order to interpret the data that we have been presented. Usually you only rely on your knowledge. But in today's, when we have a lot of omics data, the fields, every science field is shifting from knowledge-centric to a data-centric model, where the data comes first. Like you produce the data, or you observe the data. Like you observe the animal in the field, you collect like DNA from them, and you want to understand biology from those data. So computation now comes first. You analyze the data, you visualize the data, you might do some simulation based on the data. And then you try to compare the result against your knowledge. Is the result match my understanding of genetics? Does the result match my understanding of biochemistry? Knowledge comes kind of second. So we use the data to discover new patterns, and then we try to verify it with existing knowledge. And in order to be able to do the data analysis kind of correctly, we need to have some knowledge about which tools kind of does what, right? And what are the limitations, what are the assumptions for each tool. Like if you use the wrong statistical test, then you get the wrong conclusion. So it's very important that we not only know what the tools are, we have to know how it works. We have to know what's the assumption and what's the limitation. So I take this page from Wikipedia. If you ask like how many sequence alignment software there are in the world, I think you can find more than 100. So this is a Wikipedia page for sequence alignment software. And this is kind of truncated. There are many more tools kind of below this. So even with just very simple tasks, like we have DNA sequence, we want to align the two sequences together, you already have tens of options, hundreds of options. So how do you know which one is good? How do you know which one is the right one for your data? You have to have some computational knowledge. And also, when you want to talk to people who knows how to analyze data, like if you want to collaborate with other scientists, with other researchers, if you have no idea about the terms or the names that they are using, then you cannot communicate. So knowledge, computational knowledge is also useful for communication with computer scientists, for example. Okay. When we think about computational thinking, you probably approach many problems before, even in biology, in medicine, or in veterinary. You have to solve some problem. So computational thinking is a process for solving problems. But we want to do this kind of computationally. What it means is we want to this first step is the same for every approach. Like we want to talk to people who understand the problem. We want to define the problem. And we want to break them into individual tasks, individual small tasks that we can solve. Then this is where computation comes in. We want to solve this problem computationally. So we need to know which parameters are involved in the problem. Right? If we want to study cells, we want to know the gene, we want to know the mutation, we want to know the gene expression. These are the parameters that we need to study. And we need to be able to model how they interact with each other. Does this mutation affect the expression level of the gene, or does it not affect? Once we have the parameters and so this defines like how do we collect the data. If we know the parameter of interest, if it's genetics, then we do DNA sequencing. If it's on the protein level, then we need to do proteomics. But once we collect the data, then we need to analyze it and try to identify patterns. If you want to identify biomarker for a certain disease, you need to get a lot of DNA sample or blood sample from patients or from animals with that disease. And you want to compare it to other animals or other humans that are healthy. And then you want to identify gene or protein that change between the two. We want to identify those patterns that can tell us something about the biology of the system. But once you have the pattern, once you have the biomarker, then you need to test it. Like if you want to use it in the field, you want to use it in the hospital, you want to use it in your lab. The last step is to develop the biomarker that you have or the pattern that you found. Can you come up with a new assay that you can use to detect that pattern? Instead of analyzing all genes, can you analyze just a single gene that is a marker for your disease so that you can do it kind of cheaply and do it quickly? So that's the last part of the computational thinking, right? Finding the algorithms, finding the solution that you can use everywhere. When we talk about computational thinking, like in the era of AI, some of you might have the impression, even many of my friends have the same impression that, oh, now we have the very smart AI. We have a large language model. Can we just use it? does it solve everything? Basically, can we kind of stop having to worry about the details and then just kind of use it if it can give the right answer? The thing is, if it's in everyday use, or if it's something that is based on opinion or subjective topics like discussion about the politics or the news, like we can talk to large language model. It will have opinion that it kind of correct from all the training data. But in science, if we want to say something is true in science, we need to know the mechanism. We need to know why it's true. And language model mostly doesn't learn from causation. It learns from correlation. It reads a lot of text, so it knows that, okay, this concept appears a lot together with a different concept. But that doesn't mean that it knows which one caused that A is cause B. It only knows that A and B appear together a lot. When we get the results, when we get the response from this AI, we have to have some knowledge. Otherwise, we cannot verify if the response is correct or not. If you know nothing about, let's say if you know nothing about physics, right, and you ask large language model about physics, and it tells you something, we have no idea. We cannot say if it's true or not because we have no knowledge about physics. And if we want to trust what the AI tells us, then we need to know the explanation. Right? We want to ask the model, how did you come up with this answer? What is the scientific principle that kind of give you this answer? So this is a problem. Not the problem really, but it's an issue with complexity. When we have very large model, like it's in the name, like large language model. When you have a large model, you have very complex model, you can essentially capture everything. Like this quote that I got really captured this concept that if I give you 4 parameters, like 4 variables like X, Y, Z, W, then you can model the movement of, then you can model an elephant. If you want the elephant to move, you need more parameters. You can just add more and more parameters, and you can make the same elephant do many things. It doesn't mean that your model understands what an elephant is. It's just that your model has enough parameter to capture the elephant. It doesn't tell you about the mechanism, how the elephant moves. It only tells you that, okay, it needs 5 parameters if you want the elephant to move his trunk. So when we study statistics, some of you might have not so good experience with statistics when you think about it as formulas. If you have to remember this test has to be used when and where, it's not really, it doesn't involve much knowledge. It doesn't involve much understanding. So in this class, we're going to talk a lot about statistics because it will come up in many algorithms. What we're going to talk about the motivation and the models that goes on inside those statistical techniques. What I mean is when we study one statistical test, in this case, my example here, you will see it in later lectures, but when you do, let's say gene expression, transcriptomic analysis, and you want to compare genes that are differentially expressed between the 2 conditions, and then you want to understand which biological process or which biological pathway are enriched or are observed more frequently in those genes, the gene that change, either upregulated or downregulated. You have to do some statistical testing. Like, what do you mean by enrichment? And underneath that kind of simple question, there is a probability model. It's called, the name is actually called hypergeometric distribution, but you don't have to worry about the names. It describes how you can calculate if you have a certain number of genes, a certain number of genes belonging to a biological function. How does that translate to statistical significance? Or in this example, let's say we have a Thai population of 70 million. We know that, let's say we have some data that 1 million of them have spontaneous nose bleeding. So you have blood coming out of their nose without any reason, just suddenly have a nosebleed. And we know that 20 million of Thai are exposed to high PM 2.5. If you are a scientist, like you might want to study, is the nosebleed caused by PM 2.5 or not? And you collect the data and you found that there are 300,000 Thai people that have both conditions. So have spontaneous nose bleeding and also exposed to high PM 2.5. A statistical question would be, is there a significant association between PM 2.5 and spontaneous nose bleeding? I think I pulled this from some news, some news article. So if we understand how to calculate this significance, and we're going to talk about this model in this class and in a later lecture, you will know why we use a certain formula for this. So this is how we will study statistics in this class. We're going to talk about the problem. We're going to talk about the motivation, the model underneath, and then try to use it in some assignment and also in class. Okay. So if you want to study early, it's called hypergeometric distribution. So when I did my PhD, I talked to my advisor about this as well, like the problem that we kind of remember statistical tests and kind of focus on when to use kind of which test. But in real life, when you run into a new data, when you run into a new research question, you shouldn't be thinking about kind of which statistical test should I use. You should be thinking about your scientific question and then think about which statistical test match the description of my research question. So I say this kind of unwrapping can the statistics. I think some statistics is canned, like put in a can, in a sense that if you want a t-test, you open the t-test can. If you want a Mann-Whitney U-test, you open the Mann-Whitney U-test can, and then you just pour the content and eat it. But I want to kind of teach you or train you to think about statistics from the other way. We want to start with the problem. So for example, we have a patient and the healthy control with different, the patient with acute kidney injury or AKI. And we suspect that protein X might be related to acute kidney injury. So we measure the level of protein X in the blood among the patient and among the healthy control. And we can plot the histogram. Right? This is a histogram for the expression of protein X in the blood. And we want to quantify, we want to calculate, we want to be computational. We want to know if this protein X is good at distinguishing AKI patient from normal control, from healthy control. Like some of you might look at this and then, oh, the distribution looks normal. So the rule says that we can use t-test, like independent sample t-test. But if we want to look from another way, we want to know, if you want to know if protein X is good for distinguishing these two populations or not, we want to look at, we want to come up with some score. If protein X is good, if we compute the average expression of protein X among the AKI patient compared to average expression of gene X, protein X among the healthy control, and we subtract these from each other, or we may take the absolute value of the subtraction. If this score is large, that means you have a big difference in the level of expression between the two groups. So this score can tell us if protein X is good or bad for distinguishing the two populations. But this score, like some of you might notice that this score doesn't take into account that the expression of X in AKI people is very narrow, but among the healthy control, it's very broad. The variance is different. So we can improve this score further. We can divide this by the total variance from the AKI patient and the healthy control. So this new score capture both the difference between the average expression and also divided by the variability or variance of the expression. If you like the second score, then this is actually the score that is calculated inside independent two-sample t-test. So in this class, we want to approach statistics from this way. We want to start from the problem. We want to come up with ideas that kind of make sense given the problem, and then it will link to the correct or the right statistics. Naturally, you don't need to remember anything. we can pick the right test because we know what we want from our research question. Okay. And in this class, not now, but we are going to take a peek behind many popular statistical tests that you probably know, like Mann-Whitney U test or Wilcoxon rank sum test or Thai rank test. Like, these are something that you probably have seen before. We're going to take a peek underneath them and realize that they are just something that very simple and it all makes sense. And hopefully this will make you like statistics more and make you remember the test kind of more easily now that we understand what's kind of under the hood instead of just when to use what. Okay. Another aspect of computational thinking is not only to do statistical tests, but we also want to come up with computational or mathematical formula that can describe kind of certain aspect of the data. For example, if we study the microbiome, like the bacteria inside the gut of normal children and maybe obese children, these are all hypothetical, like don't take it seriously. In normal children, they might have very few taxa, like very few bacteria, different bacteria that occupy the majority. But in obese children, because of their medical condition, they might be hosting many different bacteria. So by eye, like you can already see that the data are different. But human eye is not reliable. So we want to be able to turn this data into some numbers that can distinguish normal children and obese children. For example, you might want to count the number of different taxa, the number of different species. Then in normal children, you might get something like 4, 5, 6. In obese children, you might get 20, 50. This number of taxa can serve as some score that can distinguish between obese and normal children. But we all know that the number of taxa is a very rough score. Right? It doesn't tell you about the actual abundance of the taxa. We say that in normal children, they have 5 or 6 taxa, but it doesn't tell us that one of the taxa is 46% abundance. The other is 40%. Like these two occupy the majority of the gut microbiome. So we need something more detailed, like more resolution. So we might think about the frequency of each taxa, like 40%, 45%, 10%, 5%, 2%. How can we use these proportion or percentage to come up with a measurement or score that can better describe what the normal children gut microbiome is and what the obese children gut microbiome is? Some of you might be familiar with the concept of entropy. So let's say we have 3 situations. In situation A, we have a third of the population being gray and 2/3 being blue. In B, we have a half and half, like half gray, half blue. And in C, we have 2/3 being gray and 1/3 being blue. In terms of composition, A and C are different. They have different proportion of gray and blue. But in terms of the complexity, in terms of how complex the population is, A and C are actually the same because you have 1/3 of one population and you have 2/3 of another population. It's the same 1/3 and 2/3. It just have different colors. For B, this is a 1/2 and 1/2. So what we can calculate to distinguish between A, B, and C, we can look at the distribution or the proportion of different taxa. Let's take this example, which is easier to think about. So let's say we have a different mixture of 2 bacteria, like one green and one red. So a measurement called entropy. This is a formula. We don't have to worry about this now, but the idea is with this formula, if we calculate the entropy for the 3 conditions here, the leftmost mixture have 6 green and 6 red. So this is the entropy for when you have equal mixture of green and red. In the middle here, you have more green and less red, and you have lower entropy. On the right hand side, you have all green. Every molecule is green. Every bacteria is the same. Then your entropy is 0. So this entropy score, invented by a mathematician, can tell us how pure or how mixed a population is. So if you have a low entropy, then your population is very pure. It's dominated by a few taxa. But if your entropy is high, then that means your gut microbiome is equally occupied by multiple taxa competing with each other. So with just one score, you are able to sort the complexity of the gut microbiome from very pure to very balanced. So this is something that we want to explore in this class. How can a certain mathematical formulation is very good at representing kind of biological system. Okay. I think I will skip this one. We will see this again in the machine learning class. It's another use of entropy that allow us to create a classification model, a machine learning model. Okay. Boop, boop, boop. I think I will skip this one as well. We can go back to this afterward if you want to try to construct a statistical test together. Go from the biological question and then try to construct what the test should be. And then we will realize that this is something that statisticians also use. Okay. Boom, 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 boom. Let me skip this. Okay. So we talked about statistics from a different perspective. The next topic that we are going to cover in this class is about mechanistic modeling. So statistical modeling is about probability, but it doesn't involve knowledge from biology. We are not actually modeling kind of the movement of the protein. We are not actually modeling the interaction between the genes. But from the perspective of biology, these are important as well. So by mechanistic modeling, I want to refer to this quote that what I cannot create, I do not understand. So basically, if you want to fully understand a certain system, you have to be able to create it. You have to be able to simulate it. You have to be able to reproduce it. Otherwise, you are just kind of observing the system. You don't really understand what moves inside the system. But if you are able to create it, then that means you know every factors. You know every moving part inside the system. So let's see some example. Most of you probably have learned some undergrad chemistry. So we learn about kind of kinetics in chemical reaction. We have enzyme and we have substrate, and we have a reversible reaction between the two. They can bind together or they can dissociate. We probably learned from chemistry class that we can calculate the constant for the dissociation by looking at the concentration for the enzyme and the concentration for the substrate, and then divided by the concentration for the enzyme and substrate complex. We can derive kind of this equation by thinking about what association and dissociation mean physically. If we want to have an association or the binding between enzyme and substrate, physically it means that the molecule for the enzyme and the molecule for the substrate has to meet each other in 3D space. Okay? The molecules that travel around the solution, it needs to meet with each other at a certain point in space. Otherwise, they cannot bind. If they are far apart, they cannot bind. So they have to run into each other. They have to collide into each other at a certain point in space. And that can occur with some probability. With some probability, the molecules will run into each other. And once they run into each other, then there is a certain probability that they will bind, or they will just be near each other, but not interact. And we can estimate this by saying that if we have more molecules, then we have more probability of meeting each other, more probability of collision. And the probability scales with the number of molecules. Okay? If you have twice the number of molecules, you have twice the chance of finding that molecule somewhere in the space. So that's why the association rate is calculated by the concentration for the enzyme multiplied by the concentration of the substrate, because the probability that the two will meet depends on the number of molecules, which is directly proportional to the concentration. And then the same dissociation. If you want to have a dissociation event, right, you have to have the enzyme substrate complex first. So the number of reactions that you can have depend on the number of molecules for the enzyme and substrate complex. So that's why the dissociation rate depends on the concentration or the number of molecules only. So if you know this, right, if you can derive this from mechanistic understanding, you can rederive the kinetics in chemistry without having to remember it. And this is how we want to approach modeling in this class. We want to understand the principle behind it, and then we can rederive it anytime we want. We don't have to remember anything. At least that's how I operate. Hopefully I can convince you to do the same. Okay. But sometimes the process might be very complex. Chemical reaction is very simple. It's just molecules moving in 3D, meeting each other. But if you want to model the growth of bacteria, like on the plate, so on the plate, you put food like somewhere in the plate at certain location, and the microbe moves on this plate. Like some microbe might be able to sense the direction of the food, like based on the gradient of the nutrient. Some microbe might move randomly because it cannot sense the nutrient. So we can write this kind of simulation in Python or in R, better in Python, to simulate the growth, the number of microbes. If the microbes can run into a food, then it might be able to get the nutrient, get the energy, and then multiply into from 1 to 2. So we can run the simulation. If we start with certain microbes in blue and a lot of food like on the plate in green, and let the microbe grow, run into food, eat the food, and divide into more microbes, then after a certain time period, then we can try to simulate like how many microbes will there be after an hour, after 2 hours, after a day, for example. This is something that if we know the principle behind the growth of the microbe, the movement of the microbes, then we can simulate the expansion of the number of microbes or the number of colony on the plate. Some people use this kind of modeling to understand the growth of cancer, because cancer is growing inside the body, so it has a certain limited volume inside our body. And when a cell divides, it has to compete for space and compete for nutrient with the adjacent cells. So there are people who try to model how normal cells and then certain cells inside the tissue gains the mutation that cause it to be cancerous. And cells that gain the cancerous mutation can spread more quickly. And we can do a simulation of when a certain cell spreads more quickly, how does it dominate the other cells? Right? At the end, there will be more cancerous cells than more normal cells. But how many more? We can model this by using several assumptions. Like what's the mutation rate? What's the rate of division for normal cells? What's the rate of division for the cancer cell? And what's the death rate? Like what's the rate at which normal cells are killed versus cancer? Like cancer cells will survive longer because it turns off the cell death programming. So with this parameter change, we can simulate how many more cells, how many more cancerous cells will appear after a certain time. And some people have used this to match with actual biopsy data from the patient to check whether the model can mimic what's actually going on inside the patient's body. Oops, sorry, the wrong direction. Okay. So we have seen statistical model that use probability as a basis. We have seen mechanistic modeling that use our understanding of the physics, chemistry, or nature to come up with the model. But sometimes we really have no idea, right? And this is fair. In some system, we really have no idea what happened. We can only observe it. in this case, we call it an empirical or data-driven modeling. So empirical means that we are looking at the data and we assume that this is what it is. We don't have any mechanisms to model it, but at least we have the data, so we can do something about the analysis. For example, this is not a biological system, but it's close. So let's say we want to study the waiting time. If you go to the hospital, sometimes you have to wait, like, because there are other patients also waiting for their physician. If we want to model how the patient, when do they come to the hospital, how long did they wait for the physician, this is really difficult because no one knows when how the distribution of time that someone will come to the hospital. Like, it depends on their daily life, it depends on their health condition, it depends on the cities that they are in, it depends on the traffic. It's impossible to model how many patients will come to the hospital at a certain time. But we can observe the data. Like, we can count the number of patients every day for a year, for 2 years, for 3 years. Like, then we'll have enough data to do some simulation. Like, if the new patient in the future follow the same pattern as patients in the past, then we can use data from last year to forecast what will happen this year. But we have no actual mechanism behind it. We are not model each patient as taking the bus or taking the car to come to the hospital. That's impossible. So this is what's called like a data-driven or empirical modeling. We don't have any mechanistic understanding, but we have enough data that we can still use that data to predict the future. And sometimes we have to do this. For example, when you learn in statistics class, like, you might have learned some linear model. You have learned some linear model. It allows you to put in a table of data. Like, each rows might be different cells or different patients, and the column might be data from different genes. And then you can train a model that, okay, I don't know what the interaction between the genes are, but I'm going to approximate them as single lines. And we want to classify maybe cells that are treated with certain drugs from cells that are untreated. We have no idea about the interaction between the drugs and the genes and the protein, everything inside the cells, but we approximate from the data that this might be the pattern that they follow. So machine learning is also a data-driven technique. It's based on the data that we observed. We assume no biological knowledge, and we just want to fit the data and see how it can be used for future. Another example of a machine learning model is what's called a tree model. So this kind of mimics when you, if you have a medical exam, for example, like the doctors might ask you, like, how old you are, how are you? If you are young, like teenagers, then the doctor is going to have a certain set of questions that he wants to ask a teenager. But if you are 40, like me, then the doctors might think more about kind of diabetes, high blood pressure. Like, these are not questions that he will ask or she will ask a teenager. So a tree model kind of try to figure out which question or which condition to check first. And then based on the response from that question, from that answer, what's the next question to ask? In terms of biology, this might be which biomarker to check first, like which protein to measure first. And if that protein is high, what should be the next protein that we check? If that protein is low, what should be the next protein that we check? So this is also driven by the data. We can just kind of get data from the past experiment and try to figure out which gene serve as the best biomarker without actually knowing how that gene affect the condition or how that gene respond to the drug. Like, we look at the data and we can figure out this gene is a marker for this disease. And sometimes we don't even know how this gene causes the disease. But the data suggests that it is high in patients with the disease. So this is what's called a data-driven approach. Okay, I'm gonna skip some example here. because it's too difficult. So when we do modeling, like some of you might worry that, okay, if we simulate the patient, if we simulate the cells, if we simulate the microbe, how do we know that the simulation that we make can capture the truth? Because in nature, there are many cells, there are many microbes, and they are all different. Even if we grow the same cells, grow the same cell type in the same culture media, you still get different colony. So there's some randomness, some stochasticity in the experiment that you have to be accounting for. So in this case, we can do in two ways. And these are keywords that we will touch later on. You will see the term Monte Carlo simulation. So this is a simulation that accounts for uncertainty. So instead of saying that in the next phase, the cell will divide, we will say that in the next phase, the cell will divide with a probability of 0.8. So in our experiment, if we do the simulation 100 times, 80 times the cell will divide. 20 times the cell will not divide. So we now have kind of diversity. We now have some randomness inside the simulation. So this can capture like the erratic behavior of cells, of genes, of human better than a model that say the next step this happened, the next step this happened. Another technique that we will see later on is called bootstrapping. So if we have two samples, like we have data from two patients, if we use, and let's say we do DNA sequencing, we have a lot of DNA fragments. If we analyze all the DNA fragments and get one measurement, then we all have two data points, one from patient 1, one from patient 2. And with two data points, you cannot really do any statistics. You don't know, let's say if the patient 1 has 20 DNA fragments, you get number 20. We have no idea if this 20, is it 20 plus minus 5, or is it 20 plus minus 1? Right? Because we only have one number, 20. But bootstrapping is a technique where instead of analyzing all the data at once, you sample some of the data and then analyze it. So from one patient, you can analyze it 3 times, 10 times, 20 times. From patient 2, you can analyze the data differently, 10 times, 20 times. And then you, from these many measurements, you can calculate the variance of the measurement. So now you know that 20 reads, 20 fragments, is it 20 plus minus 1, or 20 plus minus 5, or 20 plus minus 10? We will see this in several omics analysis later on. Okay, another keyword that I want you to see, this is probably new for most of you. It's a term called optimization. Optimization means you want to make something very large or very small. You want to optimize it. Why do we care about this? I want to show, and let's see if I can convince you that optimization is very important. Some of you might have fit a curve to your data before. Like when you want to draw a graph, like you want to fit a line, you have to find a line that best fit to your scatter plot, to your data point. And by best, you mean that you want to minimize the square of the error between the curve and the data point. And this is called least square fitting. Like when we want something that's best, that means you are optimizing something. Like if we want to model the drug protein docking, like how the drug binds to the protein, you want to minimize the energy of the drug protein complex because that's where it's most stable. So you're minimizing something, you're optimizing something. If you want to improve the queuing of the hospital, like in the example that I mentioned, you want to minimize the average waiting time for the patient. So you are going to optimize something. If you want to annotate cell type, like you get transcriptomic data, you get gene expression data, and you want to annotate the cell type. That means you want to select the cell type that is most similar to the gene expression pattern that you observe. So you are maximizing similarity to known cell type. And in the next lecture, we're going to talk about sequence alignment. Like when you have a new DNA sequence and you want to understand what this sequence is, like where it comes from, which gene it is, you need to align it to a database. When you do the alignment, you are essentially trying to minimize the number of mismatch position between your sequence and the database. So you're doing some form of optimization. Okay. And so how do we optimize something? Okay. Let's take the most basic example. If we fit the curve to the scatter plot, we want to minimize the error or the red dashed line here. You want to minimize these red dashed line together. I'm going to go quickly here because it's not really important. I just want you to see the idea. So you might be able to guess, like if you want to fit a line, like you have to pick the slope for the line and you have to pick the intercept. You might be able to guess that my slope, like the parameter A here, I might take the sum of all the y coordinate divided by the sum of the x coordinate, because that's the definition of slope, like y divided by x. And you might have a guess. And this is called a heuristic guess. You might use calculus. You might take the derivative and set it to 0. And then you get some answer. And this can be where the function is based. But that's not important. Just want you to know that when we think about optimization, we have probably seen many techniques for optimization already, but you might have not think about it, because I may have not thought about it this way. Okay, let's talk about this example instead. There's a famous optimization problem called a knapsack problem. A knapsack is basically a backpack where you can put things in. Okay? The problem of knapsack is it can only carry a certain weight before it broke, before it tears. So let's say you have a bag or a knapsack that can carry 15 kilograms. And you have several books that you want to carry, go somewhere and then sell it in the market. So you want to pick as much value as possible into the bag. Not as much weight as possible, but as much value as possible. Because at the end, you are going to sell the book and you want to get as much money as possible. You are not focusing on getting the heaviest book, but you want to get as much value as possible. This is called combinatorial optimization because you have a lot of ways to pick the books. You can pick this or that. This might seem kind of out of biology, but actually if you think about evolution, evolution of an organism, it's a combinatorial optimization of the genome. When we think about the genes, each gene has different value. Like if we insert a certain gene into the genome, it has a certain function, so it adds a certain value to the genome. But if you have a large genome, like with many genes, then you also have high maintenance because each replication, you're going to have to clone all the genes. So evolution is a trade-off between getting more genes and having more value versus having few genes and having better maintenance, lower maintenance. So this is the optimization kind of thinking happen in nature as well, and it will come into biology in many ways. Okay, so in biology, so let's see kind of how we encounter computational thinking in biology. First, some of you might already thought about which keyword to search in the registrar, like when you want to find computational biology or bioinformatics class. Like you found my class probably through either some search of computational terms. So in biology, there are many fields that approach the problem computationally. Computational biology is a direct one. Like we ask biological questions, but using computational tools to analyze. Some people define themselves as system biology, so they don't care whether they use wet lab or use computational tools. But what they think about biology is that it's a system. It's not about origin. It's genes, gene interaction, epigenetics, protein regulation, and then environmental impact. Like, cell is not existing alone in vacuum. It has to interact with many things. And then we have bioinformatics, where it focuses mostly on algorithms. How do we optimize the sequence alignment techniques? How do we make it faster? How do we make it more accurate? The focus is on the algorithm. So this class, to show why this is our expectation, we are computational biology class. So we will touch a little bit about algorithms. We will touch a little bit about system thinking, but we mostly want to answer biological questions using any computational tools. Okay. And if I ask you, like, go back like 20 years, 30 years, probably some of you haven't been born yet, but back then, not many people in biology do computation. Like, and it's not because they don't have computers. It's because they don't have the data. So the Big Bang moment in biology, if I want to ask you, like, what do you think is like the birth or the Big Bang moment of modern biology? What would be that moment for you? You probably are too young to answer, but so basically it's the moment that we have the first human genome. And the reason why, if you know about BLAST, like this is a sequence alignment tool. If you know about exome sequencing, like some of you from genetics might know, so we want to sequence only the gene, only the protein coding part. We don't want to spend a lot of sequencing effort on the non-coding, which is more than 80%, 90% of the genome. And some of you might know about the microarray technology, where you can measure gene expression on a chip. These three techniques, the one thing that they have in common is that if you don't know the human genome, they don't exist. Because if you don't have the human genome, what would you search against? Like, you don't have the database for BLAST. If you don't know the human genome, how would you be able to enrich exon? If you don't know what the exons are, you cannot generate the hybridization probes if you don't know the target sequence. You cannot create microarray, because microarray, you have to know the sequence for your RNA, so that you can decide the reverse complement, the reverse of the RNA sequence and binds to it. If you don't know the human genome, none of this technology can be made. And if you go back to kind of that period, so this was 20, 30 years ago, this was when I started university. So I was kind of lucky to be in that wave. So in 2000 to 2003, this is the period where we start to have a good enough DNA sequencing technology that we can sequence almost the entirety of the human genome. We have DNA sequencing technology for a while, but it only works for small, short DNA fragments. It's not powerful enough to collect data from the entire genome and assemble them together. So that's like the Big Bang moment for all the biology that we're going to study in this class. And if some of you come from proteomics, I came from proteomics. In proteomics, the technology, the mass spectrometry technology, has been around for hundreds of years. But it's only applicable to biology in 1980. And we will learn about this a little bit, why it's so. But I want you to realize that most of the techniques and most of the development that we have today only occur less than 30 or 40 years ago. It's all very new in the time scale of biology. But now that we have these tools, we call it a digital transformation of biology. We go from qualitative experiment, like where you look at the gels by eye. Now you have look at the gels with digital microscope. You can quantify the intensity. You can look at genes. You can look at all the genes at once, not just a few genes that you select. And you can monitor gene expression throughout the time, not just at a single time point. So this is a true digital transformation of biology. And once you have these large amount of data, then you need to have tools to analyze them. Like the first that we talked about is statistics. And more that we will talk about will be about bioinformatics algorithm that we use to understand how do we know that the Tata box is a Tata box. How do we know that it's a TAA, TTA? There must be some experiment that get the data and some algorithms that analyze data and tells us that wherever the protein or the transcription factor bind, it has to be a TAA, TTA motif. That information must come from some kind of sequencing technology, followed by some bioinformatics analysis. When we see AlphaFold today, I might go over the time a little bit, maybe less than 5 minutes. Okay. So if we look at the AI today, like AlphaFold, that can predict 3D structure for the protein, if you go back in time and look at the AlphaFold version 2, AlphaFold version 1, and then a few years before AlphaFold version 1, you will see that there are already attempts that try to model protein 3D structure, but using evolutionary information. And we'll talk about this later in the structure, protein structure lecture. But I want you to kind of appreciate that the development that we have today kind of comes from many moving parts, many foundations that were made kind of earlier. And each of them we can understand. It's something that you can understand. But nowadays you might see them packaged in a very advanced AI. And it's difficult to distinguish the ingredients kind of inside. Okay. In a lecture, in one of the lecture, we will learn about what's called a network or graph, where we connect kind of gene and protein together if they are interacting inside the cells. And from this kind of representation as a graph, as a network, we can make a lot of interpretation, like which protein is important. What's the definition of importance on the graph? Like, do we want a lot of interaction, or do we want the protein to connect other protein? What does it mean by important in terms of biology? And how do we transform these two statements into mathematical or computational calculation? Okay. And this is we can talk about this later, but people from different backgrounds, like electrical engineering or civil engineering, can approach biological problem using their knowledge, using what they are familiar with. For example, my friend, when I was in undergrad, she is an electrical engineer. So she modeled gene interaction, like how gene interact inside the cell, as an electrical circuit. And she can calculate how much information flows across the gene by treating the flowing information as an electric current inside the circuit. And because most of you probably have studied circuits in high school, you know that if we know all the resistance, we know all the entire circuit, we can calculate the amount of current through any wire inside. Okay, so we have seen statistics, right? And it's important in how we distinguish between data and the pattern in the data, which one is significant. Right? We have seen algorithms that allow us to identify patterns, like DNA binding motif, or when we do the sequence alignment with BLAST, we need to have some algorithm that says, this sequence is similar to my input, and this sequence is not similar to my input. In the next lecture, we're going to talk about how algorithm like sequence alignment join with statistics to tell us which alignment is significant, which alignment is not. Okay. So this year we're going to have 4 modules, so I can go over this quickly. The first module will be about DNA sequencing, about genomics, metagenomics, mostly focusing on DNA. The second module is going to focus on gene expression. We're going to talk about why transcriptomics is very popular and what can we do once we go from bulk transcriptomics to single cell or spatial transcriptomics. For the third module, we're going to cover broad fields in computational biology, from proteins to dynamics modeling, to networks, to structure and imaging. So if you have seen the content from previous year, you will realize that this year the shift kind of changed from doing a lot of Python in class to giving you more coverage of the fields in computational biology. And in the last module, we're going to touch on the machine learning and AI that there's no escape. Basically, you're going to run into machine learning and AI, so you might as well learn it from your first year with me. Okay, so we're going to talk about some application with machine learning and AI in biology. Okay, so I already talked about the assignment. So you will be analyzing some data. You will be reading some papers and extract some key details. You will be designing some experiment using the tools and the technique that we learned in class. And we will be critiquing, so discussing about what large language model produce. Okay. And yeah, if some of you want to edit some, debug some R and Python, that will be there as well. Okay, so if you have to go, please feel free. But if you have any question, I will remain in here to answer.